In the last lecture, we spoke about the formation of enamel, that is amelogenesis, and we spoke about the life cycle of ameloblasts, the electron microscopic appearance, the enamel rods that are present, how they are formed, and the different. Today, we will be discussing about the structure of the enamel, the physical chemical properties, as well as the light microscopy and electron microscopic appearances of enamel. Enamel is the hardest tissue in our body. It accounts for 343 kHm. K stands for Knoop hardness number. Because of the excessive amount of minerals that are present, it is a very hard substance. But on the contrary, because it does not have enough organic content, it is very brittle. So very easily it will fracture and break down. It has a specific gravity of 2.8 and it is very translucent, giving a grayish color. So when you see enamel, you are actually seeing the dentine color through the enamel. The thickness varies from different parts. It is around 2.5 millimeter at the posterior cusp tips, and at the cement to enamel junction, they are feather edged. Enamel is not totally impermeable. It allows diffusion of molecules. It is an important factor because of the permeability, it allows fluoride to get incorporated, which has the tendency to make the enamel more hard. Enamel, as we have discussed in the last lecture, is 96% inorganic. This 96% inorganic content is mainly hydroxyapatite crystals, along with some other minerals like magnesium, lead, and of course fluoride. These crystals are hexagonal in shape and have very less amount of inter-rod substance that is predominantly tyrosine-rich amelogenin protein and a little amount of leucine-rich amelogenin protein. The structure of the enamel is basically enamel rods. As you can see from the surface of enamel down to the dentinoenamel junction, you have rods that are traversing. These rods are formed by the hexagonal hydroxyapatite crystals. These rods are aligned one above the other. And the unique feature about these enamel rods are that these rods do not follow the same line. They are curved in all the three dimensions of x, y, and z axis. If you observe here, the first rod and the next adjacent rod are offset by an angle of 2 degrees. So if this is at an angle of 4 degrees, the next rod on top will be at an angle of 6 degrees. And this pattern proceeds throughout the structure of enamel, giving a sinuous appearance on the surface. So it not only goes in one direction, but also goes in the opposite direction, giving rise to a wave-like pattern. So there will be depressions and elevations of the direction of the enamel rods. Because of these depressions and elevations, you can actually appreciate that when a cross section of enamel is taken, you can see that there are some rods which are sectioned cross-sectionally and some which are sectioned longitudinally. This gives an alternating longitudinal and cross-sectional appearance on the enamel. Let's have a look at the enamel rod pattern. If you look at the cross section, you can appreciate the keyhole pattern. This keyhole pattern, when we section it in sagittal, coronal, and transverse sections, you can appreciate the direction of the enamel rods. Now, having a look at the B section, that is the longitudinal section, you can appreciate that the central portion of the enamel rod have horizontally oriented rod crystals, whereas the periphery, that is the inter-rod substance, are angulated at an angle of 60 degrees roughly. When you look at the sagittal section, you can appreciate that the enamel rods are angulated, and this is also roughly forming an angle of 60 degrees because it is the inter-rod substance. On cross sections, you can appreciate the keyhole pattern with the central head of the rod and the peripheral rod sheath. We need to understand this architecture of enamel. The rod and the interrod meet at an acute angle, and this acute angle formed allows some amount of organic matrix to be present. So this area of the rod and the interrod region will have the highest content of organic material, which is amylogenin proteins. And this organic content can be easily leached out. So if a bacteria sits on top of this particular enamel rod and leaches out acid, the very first area that gets dissolved is this interrod substance. Once the interrod is dissolved, the rod actually chips off, giving rise to a cavitation, which leads to caries progression. These rod structures are roughly 5 microns wide and 9 microns long. They are of variable width and is wider at the middle. The rodless enamel, which is the outermost as well as the innermost surface where the Tomes process is not formed, is around 30 microns in thickness. When the amyloblasts are depositing enamel, they take rest on a daily basis. So a diurnal rhythm of the amyloblast during the rest phase leads to a excessive amount of organic material being deposited at a frequency of 4 microns. These are called as cross striations. Similarly, a circumseptamanian rhythm, that is a weekly rhythm, is also noted, which is called as the incremental lines of Retzius. So if you look at this image on the left, you can appreciate that the enamel rods have an even increments of smaller duration, that is roughly 4 microns. These are the cross striations, which can be better seen on the right hand side image as well. On the other hand, the striae of Retzius is much more larger duration. So if you look at this particular image, you can see that the enamel rods are traversing from the surface to the dentino-enamel junction, and perpendicular to those are the daily cross striations. These are the daily cross striations. Oblique to the enamel rods are the ones where you see the striae of Retzius. These are concentric lines which are formed by weekly rest-taking phases of amyloblasts. These concentric rings are more appreciated in a longitudinal section. So here you can appreciate the concentric rings that are formed. And if you observe keenly, there is accentuation of a single striae of Retzius. These striae of Retzius are because of a change in the physiological condition during childbirth, and hence called the neonatal line. So what happens in a neonatal line? During the child's development, intrauterine, the enamel is formed in a very controlled environment. This controlled environment is provided by the mother's womb. And during deposition of enamel, the enamel formed, that is the first formed enamel, is very uniformly deposited. The striae of Retzius are very uniformly spaced. So the very first formed enamel, that is closer to the dentino-enamel junction, you can see that the striae of Retzius are evenly spaced. At childbirth, the child comes out into the environment where he has to fend for himself by eating food. The liver starts to act and has to undergo the process of digestion, etc. And the nutritional supply is slightly altered compared to the intrauterine maternal circulation. This leads to an accentuation of the resting phase of the amyloblast, giving rise to an accentuated striae of Retzius called as the neonatal line. Now this neonatal line, when you look at it, can be used for different purposes. Also, age estimation. In case of infanticides, you can actually look at this neonatal line and try to identify the time of death of that particular child. Neonatal lines are always present in all the deciduous teeth, but in the permanent dentition, it is present only in the permanent first molar because that is the only tooth in which the mineralization occurs at birth. All the other permanent teeth, all the other permanent teeth will have formation of enamel after the birth of the child. So what exactly happens? This is a diagrammatic representation of ameloblasts during the deposition of enamel. This is a very squarish looking Tomes process. In reality, it is conical, but for diagrammatic representation and ease of understanding, it has been depicted as a rectangle. This rectangular process here, which is depicted, is the Tomes process, and the rods are deposited at one angle at the tip and at another angle at the interrod region. During the time of rest, this Tomes process constricts, giving rise to an extra amount of interrod substance being formed. This extra amount of interrod substance indicates extra amount of organic material, hence it is a hypocalcified structure of enamel. Both cross striations, striae of Retzius, which is a variation seen as the neonatal line, all three are hypocalcified structures of enamel. These hypocalcified structures of enamel, that is the striae of Retzius, are concentric as we discussed. This concentricity ends at the surface of the tooth, and the surface of the tooth also shows these striations, and these are called as imbrication lines of pickerel or pericymata. The pericymata gives rise to an irregular surface of the enamel. The smaller elevations are called as enamel caps, and the larger elevations are called as enamel brocks. This is the same image that I showed you previously, which shows the cross section as well as the longitudinal sections of the enamel. This alternating cross and transverse section, when you see it under reflected light, it gives rise to an alternating light and dark bands. This alternating light and dark bands that you see is because of the elevations and depressions of the enamel surface, and these are called as bands of Hunter and Schreger. The dark bands are called as the parazones, and the light bands are called as the diazones. An easy anagram to remember this would be the second alphabet of dark and the second alphabet of para are the same, second alphabet of light and second alphabet of dia are the same. The Hunter-Schreger bands, when you see it under the reflected light, you can see the alternating light and dark bands. It is because once the light is shown from one particular direction, the elevations get lighted up, whereas the shadow of the elevated surface falls down as a dark zone. So hence we see light and dark bands alternatingly. This does not extend from the dentinoenamel junction to the surface, but generally is seen in 4/5 of the thickness of the enamel. When you see it under a transmitted light, you see the same image of alternating cross and longitudinal sections. So till now we have spoken about the physical and the chemical properties of enamel, the enamel rods, the shape, the undulations, the striations, and the Hunter-Schreger bands. Next, we will discuss about the different structures that you see in an enamel, namely gnarled enamel, enamel tufts, enamel lamellae, enamel spindles, and dentinoenamel junction. Let's look at gnarled enamel. Gnarled enamel is seen at the cusp tips. So at the tip of the cusp, the enamel rods, instead of traversing straight from the dentinoenamel junction to the surface, they get intertwined with each other. These intertwined enamel rods make the enamel a little more harder, which is required at the cusp tips. So the presence of gnarled enamel actually is a functional adaptation which allows higher forces to be tolerated at the cusp tips. The permanent and the deciduous teeth have differences in the direction of the enamel rods. In the permanent teeth, we have the enamel rods directed perpendicular to the dentinoenamel junction and travels straight to the surface. And it keeps on happening throughout the cervical portion except for the cervical most part of the enamel, where in permanent teeth, the enamel rods are traversing apically. That means the enamel rod at the dentino-enamel junction is more coronally placed than the enamel rod at the surface, which is more apically placed. This does not happen with the deciduous dentition. There is a clinical relevance to this. So whenever you are preparing a cavity, you may have to use a gingival marginal trimmer at the edge of this particular cavity preparation so that the enamel does not fracture. This has to be done only in the permanent teeth and need not be done in the deciduous teeth. Another hypocalcified structure of the enamel is the enamel tufts. The enamel tufts are deriving their names based upon the appearance. The enamel tufts appear like a tuft of grass, and they appear to be originating at the dentino-enamel junction and extending into the enamel, not too much to an extent, maybe 4 to 5 microns above. But what is it in reality? These are actually cracks. These cracks are not present at a single location. So to see that analogy, let's section enamel in various levels. In the first level, let us say there is one single crack. Just above this crack, if there is one more crack in a different direction, but occurring at the same origin or maybe nearby origin. So if you keep on accumulating the different layers of enamel, having cracks originating from the same anatomical site, you will observe a tuft-like appearance of enamel hypocalcified structure. These tuft-like structures will be visualized as a tuft of grass like this. Another hypocalcified structure is the enamel lamellae. Contrary to the enamel tufts, enamel lamellae are leaf-like structures originating from the surface of the enamel. Enamel tuft originated from the dentino-enamel junction. These are also geologic faults. When you have a very brittle substance and you are applying a lot of force, it is having a tendency to fracture. Now these geologic faults that you see on the enamel allow some movement of the enamel to a certain extent so that it does not fracture. They may extend from the surface to the depth of the enamel to various levels, and based upon how it has been formed, it has been classified into three types. The first type is a poorly calcified rod. The second type are degenerated cells. The third type are cracks filled with organic matter from saliva. Let's have a look at these three entities. So if you look at the poorly calcified rods, the problem here is the amyloblasts are alive but are functionally not producing enamel. So when there are amyloblasts present, they move backward but they do not form mineralized enamel rods. So you see poorly calcified rods. The second type is when amyloblasts are present but they die, and the dead amyloblasts neither go backward neither deposit enamel, which leads to a geological fault containing dead cells which are nothing but amyloblasts. The third type is unique. The third type is after the enamel has been completely formed because of a trauma or a traumatic incident. The enamel cracks, and this particular type of crack can extend from the surface passing through the whole length of enamel and even into the dentine. These faultings blocks the excessive forces or strains that are produced on top of the enamel, preventing further fracture of enamel. Another hypocalcified structure is the enamel spindle. The enamel spindles are actually at the dentino-enamel junction, extending into the enamel in the form of a small club-like structure. This small club-like structure is actually an extension of the dentine. So what is inside the dentine is the odontoblastic process. The odontoblastic process enters into the enamel and gets entrapped and gives rise to these enamel spindles. The dentino-enamel junction is not a hypocalcified structure. It is a hypermineralized zone, and it is appearing in the form of a scallop. And this scalloped appearance is very unique because the amyloblasts are actually interacting with the odontoblasts in this particular zone. The dentine forms first, followed by the enamel. When the dentine first forms, there is a layer of collagen that is left out unmineralized at the dentine enamel junction. These unmineralized collagen is the initial site of mineralization of the enamel, holding onto enamel and dentine together at the dentino-enamel junction. Following the deposition of the full length of the enamel, the amyloblasts, before they finish their properties of deposition of enamel, they lay down a thin organic membrane on top of the enamel. This is called as the enamel cuticle. This generally happens in the protective stage of ameloblast, also called as Nasmyth's membrane. During eruption, it may be stained by some bacteria and appear green in color. This is nothing but a basal lamina or a basement membrane-like material. As soon as the tooth erupts, this particular enamel cuticle dissolves or is abraded away and is immediately replaced by a secondary pellicle, which is derived from saliva. Let's look at the age changes that you see with enamel. Enamel is unique because the amyloblasts that form enamel are no longer available after the eruption of the tooth, making enamel a non-regenerative material. This non-regenerative hard material tends to get atreated because of the physiological forces that are applied by the process of speaking, chewing, and holding on to things with the teeth. That is called as attrition. In a normal individual, as the age advances, as you eat, as you speak, as you have habits, the attrition rate is roughly correlating with the age and allows age estimation processes in forensic techniques. The teeth also tend to darken with age. The one reason for this is the increase in the organic content of the dentine, making the dentine a little more yellower. Not only that, the thickness of the enamel is undergoing attrition and becomes thinner, making more of the dentine visible through the enamel, making it more yellow in color. The permeability reduces with age, and the fluoride ions tend to increase on the surface of the enamel. So why do we have to understand all these features? The very first feature that you should remember is the enamel rod direction. The direction of the enamel rods are divergent at the cusp tips and they angulate at the cervical portion in the permanent teeth. Hence, during the cavity preparation, you should always go cutting in the same direction as the enamel rods, preventing unsupported enamel rods. If you have a restoration with an unsupported enamel rods underneath, the unsupported enamel rod may break down, giving rise to secondary caries. The grooves and fissures on the occlusal surface of the molars and the premolars are weak spots, and very easily food can get entrapped in that particular region. This gives rise to excessive amount of bacterial invasion in that region, giving rise to acids which degrade the enamel, giving rise to caries. Use of pit and fissure sealants, especially in deciduous dentition, have been proven to prevent progress of caries. Not only that, each enamel rod has a rod sheath around it. The rod sheath is less mineralized compared to the other structures. So the rod sheath will be the direction of spread of caries because it allows the penetration of the acids produced by the bacterial organisms. The striophragous tufts, spindles, they also may facilitate the spread of caries because these are all hypocalcified structures. Fluoridation decreases caries mainly by replacing the hydroxide ions in the hydroxyapatite crystals and replacing it with fluoride. Fluoride is a much bigger molecule sitting in a very cramped up hydroxyl group region, making the enamel very hard. But excessive of fluoride also gives rise to fluorosis, which will make the enamel weaker. Can remineralization occur? Yes. When you look at saliva, saliva is rich in calcium and there are proteins and enzymes like histatin. Histatin, along with fluoride and calcium ions, have a tendency to remineralize the surface of the enamel. Later on, we will discuss about caries in which the surface of the caries in an enamel are known to show higher mineralization capacity. The defects in the amylogenin gene, especially linked to the X chromosome, are known to cause a disease called as amylogenesis imperfecta. There are various defects in the amylin gene also noted, which are known to cause autosomal dominant type of amylogenesis imperfecta. In the process of restoration of the teeth, there are times when we will have to stick something to the surface of the enamel. So when you put a layer of acid on top of enamel, you will appreciate that the enamel differentially dissolves. The rod sheath and the rod differentially resolve in different conditions. So if you look at the first image here, the first image, the rods are disappeared and the rod sheath are intact. In the second image, the rod sheaths have disappeared and rods are intact. In the third image here, it is haphazardly dissolved. And in the fourth image, you can see both the types of A and B. So what does this provide? This provides some irregularity to the surface of the enamel. This irregular surface allows more physical surface area to be used for holding on to material like composites and cements. Hence, we have discussed enamel under the headings of formation, that is amylogenesis, under light and electron microscopy. We have discussed the properties of enamel, both physical and chemical properties, and we discussed about the hypocalcified structures of enamel along with an optical phenomenon called as the striae of Hunter-Steger bands. And then we have also discussed about the hypercalcified structure of dentino-enamel junction, a note on age changes of enamel, and clinical considerations. Thank you.